One of the things that we did talk about in class already is the semi-conservative nature of DNA replication. We understand that the parental DNA molecule is going to act as a template, the strands are going to separate, and that we are going to build new strands of DNA on the old strands or parental strands, and that the new daughter molecules will contain one old template strand, one parental piece, and one new strand of DNA that is made during the process of replication. We're also aware, because of our work on chromatin packing in the nucleus of the cell, that there's an awful lot of DNA in there, and we understand that replication cannot occur in a eukaryotic cell on all of the DNA at one time. If it was all unwound to be pulled apart and used as a template, then our cells would be blowing up all over the place because there is just so much DNA material in the nucleus. So we have to open up little pieces of it at a time and replicate it and then rewind it around those histone proteins that we talked about so that we can keep our cellular structures intact. It's good to take a minute now and think about when this process is going to occur. And these pictures here should look familiar to you. These are pictures of mitosis occurring in probably in onion cells. And so what I want you to remember is that the purpose of replicating DNA is in fact in preparation for cell division, and that because the cells are going to divide, 2 copies of the DNA are required. That holds true, pardon me, both for mitosis and for meiosis. So we understand that a second set of DNA must be made. So we're going to break the process of DNA replication down into 3 main steps. First, strand separation. How do we get that DNA strand pulled apart? Next, how do we build the new strands of DNA using the original strands as templates? And also we're going to look at how errors are going to be fixed if they occur during replication. So starting with strand separation, as mentioned previously, there has to be multiple origins of replication. So we've got specific points that are going to be the beginning points for replication to occur along various strands of DNA. And there's a special enzyme complex called helicase that will bind to the origins of replication. And when they bind, they will start to break the hydrogen bonds that occur between the complementary base pairs in the DNA strand. So if we can imagine from this little diagram here that helicase is our yellow arrow, it's going to start breaking the bonds between the 2 DNA strands that we see here in blue, and the bases, which are in the multiple colors, breaking through those black dots, which are going to represent the hydrogen bonds. As those are broken open, we're going to form a structure that looks a bit like a Y that we're going to call a replication fork. There are some issues with this, so it seems pretty straightforward. Helicase breaks hydrogen bonds between complementary bases, splitting the DNA molecule into a structure which we call the replication fork, and that happens in multiple places on each DNA strand. There are a few challenges that we need to overcome. Number one is, think about taking a rope and separating the strands of the rope in the middle, and then pulling those apart. And what happens at the ends is that you get tension building up, so it kind of bunches up. And we really don't want our DNA to be bunching up. So what happens is that The cell employs a group of enzymes called topoisomerases. Sometimes you'll also hear the word gyrase in there, so you may want to write that in on your sheet as well. These enzymes are going to relieve the tension in the DNA strand by cutting the phosphodiester linkages in the strands near the replication fork. Sometimes one strand will be cut, sometimes both strands will be cut. And because, or as a result of this, we are able to relieve some of that tension. So we unwind and then rejoin the phosphodiester linkages. So that helps with that first issue. The second issue is that those bases are pretty happy to be joined in those hydrogen bonds, and because those bases are just opposite each other on the DNA strands, then as soon as we break them apart with helicase, really they kind of want to stick back together again. And that idea is called annealing. So our bases would like to re-anneal once they are split. So we need to have something to hold them open. And that something is a group of proteins, which we call single-stranded binding proteins. And they are going to help keep the DNA strand apart. In our diagram here, we can see we've got DNA helicase in this wedge shape here. We've got topoisomerase, which is going to cut and relieve the tension, sort of spin it a little bit for us, so we can continue to move the DNA helicase enzyme complex along. And in purple here along the outside, we can see that we've got single-stranded binding proteins, which are going to help hold the strand apart temporarily. We're going to be removing and reusing those along as we open up along this replication fork, but it's going to hold it open long enough for the next part of the process to occur. So something that probably has occurred to you is that if we're starting with a replication fork like this piece right here, and we are moving, replication is occurring in this direction. Also, helicase is another helicase complex will be opening in this direction. You can see that what we end up with when we have two forks together here is something that's called a replication bubble. So helicase works in both directions from the origin of replication. It will open up, and soon the new strands will be built, and we'll talk about how that works in a few minutes. And eventually what will form is a replication bubble, and finally those replication bubbles will become so large, as we can see sort of down here on this strand, that the bubbles will meet up with each other, and eventually a whole strand with no gaps in it will be formed. Okay, something kind of neat to think about is the speed at which this is taking place. So in grade 11, you learned a lot about mitosis and meiosis, and about the fact that it's part of the cell cycle, and that in interphase we have the doubling of the chromosomes, but we don't really talk about how fast that is. So in eukaryotes, it tells us here that we can build about 50 base pairs per second at each replication fork. That's pretty quick. However, if there was only one origin of replication, it would take about a month for us to replicate the DNA in the cell, which is, is way too slow for what needs to be going on. Our cells would then not be able to carry out their regular functioning. Because we use multiple origins, we can do a complete copying of a eukaryotic genome inside the nucleus of one cell, and would happen in many cells probably at the same time, in about an hour. So it's a very effective and efficient way to get our replication done. Now that we've got our replication fork open, we're going to have a general overview look at how we're going to add nucleotides in to create our growing strand from the template or parental strand that we have. So if we look at this diagram, we can see that we've got a template strand, which we can see right here. This would be our template strand. This template strand is oriented at the top 3', at the bottom 5'. And we can see our growing strand, the strand that we're growing, is moving or is growing in this direction. So we have an enzyme that we're going to look at in a few minutes called DNA polymerase, and that is a name that should make the function pretty easy to guess. DNA polymerase is going to be adding in nucleotides one at a time. The growing strand is going to be growing from 5' at the top here to the 3' at the bottom. So the two strands will end up being anti-parallel, as we discussed in Unit 1. Just in case you can't remember, although you should be able to, we orient the molecule, or we look at it from the perspective of which carbon we're looking at on the carbon molecule. So if we look at let's say our template strand here, we can see we've got carbon number 1 attached to this adenine here. So carbon number 1 is attached to the nitrogenous base. Then we've got carbon 2 right here. And because this is DNA, there is no oxygen group on there. Carbon number 3 is this one with the OH group attached to it. Then carbon number 4 is attached to carbon number 5, which is hanging off our arm, I guess, if you like, and is then attached to the phosphate group. And same thing here, we've got carbon number 1, 2, 3, 4, and 5 off on this arm. So we can see that we're adding our new nucleotide, so this new in fact, this is not a nucleotide at this point, but it will be. We're going to be adding this in so that the 5' carbon right here is going to be attaching to the 3 carbon of the base that's already, or of the nucleotide that's already in here. So back to this future nucleotide, we can see that it looks like a nucleotide. It's got a cytosine base, it's got the deoxyribose sugar, it's got, oh, here's the difference. Instead of just one phosphate group, it has 3 phosphate groups. So this is a triphosphate molecule, a nucleoside triphosphate. When DNA polymerase adds this molecule onto the 3' end of the previous nucleotide, it cleaves 2 of these phosphate groups off, and then these, which is pictured right here, these phosphate groups themselves are then going to be cleaved as well, and the energy from that is going to be used by DNA polymerase to stick the molecule onto the growing strand. So we can see here it is as a nucleotide attached to the growing end of the new strand. So DNA polymerase 3 is the molecule. We have several DNA polymerases. In eukaryotes we have 3, and in eukaryotes we have usually 5, and in prokaryotes we usually have 3. We're sort of talking a little bit more about prokaryotic replication at this point because it's a little simpler. The ideas are similar to eukaryotic. So DNA polymerase 3 is going to add the new nucleotide onto the growing strand at the 3' end. And as mentioned, it's a triphosphate as it's being brought into the into that previous nucleotide. The DNA polymerase is going to cleave the 2 phosphate groups off, and that is going to be used as an energy source for actually creating the phosphodiester linkage, which is going to occur in here. Right? We need to create a new phosphodiester linkage to join that new nucleotide to the growing strand. Now that we understand that in order to add new nucleotides to our growing strand, that we need a 3' end available for DNA polymerase 3 to add to. Maybe you've already sort of thought about what one of our issues might be in the sort of initial stages of the replication process. So hopefully you already thought about this. When the fork first opens, there's nothing there. There's no 3' end for DNA polymerase to attach to. So to overcome this obstacle, what happens is that an enzyme complex called RNA primase is going to read the template strand that's open, and it's going to build a small piece of RNA called an RNA primer. So it may be a little weird to have RNA in a DNA molecule, but we will be replacing it later with DNA nucleotides. So this RNA primer gives us a 3' end that we can attach to. And if we have a look over here at the diagram, we can see that replication, the fork is opening in this direction. Primase is represented here in pink. And the primase, we don't see the one that's on this this other strand. We don't see the one over here, but primase is going to build a couple of primers, and here we can see this one is running 5' here to 3', so we can attach some DNA nucleotides in this direction on this particular strand, and our primer on the other strand was probably back here somewhere. And we're going to be able to add our new nucleotides in this direction because the 3' end is down here. And replication is going to continue in this direction. After things move along and this replication fork has opened up farther along, there's going to be other DNA polymerases other than DNA polymerase 3. In this case, we're most likely going to see DNA polymerase 1 is going to be moving along and proofreading the strands that were made. It's going to remove the primers and it's going to replace them with proper DNA nucleotides, so our final product is not going to have RNA in it. And then we've got another enzyme that's going to come along and reform the phosphodiester linkages. It's called DNA ligase, and it's like the glue that's going to hold things together. So another issue that that previous diagram may have made you think about is that our DNA strands that are building are not building in the direction of the helicase necessarily. So that's going to cause another little problem for us here. So have a look at this diagram, and I just want to point out to you that in this particular diagram, you should always check this. In this particular diagram, replication is occurring in this direction. So we can see on this top strand where the original DNA is in blue, baby blue, we can see that we had a primer that came in here because we can see our primers are in red. The primer was laid down here so that at this end a 3' was available, and then DNA polymerase 3 came in and added the nucleotides in order down in this direction. So you can see that it's actually growing in the direction that it's opening. So helicase would be opening this strand in this direction, cutting the hydrogen bonds, and as it opens up, DNA polymerase 3 will add in the nucleotides in a continuous fashion, just one after the other, after the other, after the other. However, on the other strand, maybe you can see that this was one of our primers, and on this side we can see the original strand is green. This RNA primer, hopefully you can tell, was 5' here, so that a 3' end is available here. And remember, DNA has to be, new DNA is always built 5' to 3'. We have to have a 3' end to attach our new nucleotide to. So this is the 3' end down here, and the nucleotides are going to be added in this direction by DNA polymerase. So do you see the issue here? We are opening in the opposite direction to the building direction on this strand. So what must happen is, as the helicase enzyme, again, that's right here, this helicase enzyme opens up in this direction, we can get enough space, I guess if you want to look at it that way, to stick another primer in with a 3' end exposed right here, and we can build another little segment. So we have special names for these new building strands. One of them is going to be called the leading strand, and one is going to be called the lagging strand, and you may already know which one is going to be which. In the leading strand, DNA polymerase is able to build continuously from wherever our replication origin is, from the original RNA primer in the direction that the fork is opening. It can be continuously built. Once it's got a primer to start from, it can keep on going as long as the helicase opens. However, on the other side, the lagging strand is going to be built away from the direction that the fork is opening. So like our bottom strand here, we can see it's going to have to be built in little pieces. And these little pieces are called Okazaki fragments, after the scientist who discovered that this was what was going on that other strand. And those pieces will continue to be built until we hit the next replication fork or bubble. And what will happen is that just like we already mentioned in the previous slide, our DNA polymerase 1 will go along and will find those RNA pieces and then put DNA nucleotides in, and the ligase will seal up the phosphodiester linkages to make a continuous piece. So just one more time, the leading strand will build continuously in this direction. The lagging strand will build in sections in this direction, opposite to the direction that the fork is opening.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
So once we get to the end of the process, or even in some cases as the process is occurring, we need to fix any mistakes that may have occurred in replication. When we think about this, 50 bases per second, that's very, very quickly that we're building this, and rarely do we have mistakes. So we must have a pretty good system in place for fixing errors. Initially, and most often, any errors that we have are going to be detected by DNA polymerase as it's moving along. So if you have a look at the diagram that we have here on the side, if there is a base that's put in by accident, so it's the wrong base, like they're putting in just here, the base pairing won't be correct, and so you end up with a little bit of a bump in the structure, the physical structure of the DNA molecule. And DNA polymerase 3 generally will not move on. What it will do is it will take out the wrong base and it will put in the correct one so that hydrogen bonding can occur and the DNA molecule is nice and straight. So generally DNA polymerase 3 is able to do that. Sometimes, however, on rare occasions, those mistakes may be missed as the DNA polymerase 3 is progressing. And what happens is we send, along any new DNA strand, we send what we call a repair complex. And the repair complex has a lot of proteins and enzymes, including DNA polymerase 1, which we mentioned already, and DNA polymerase 2, both of which can detect these errors and can take out the nucleotides that are incorrect and put the correct nucleotides in, seal them back up with some ligase, and then the DNA is good to go. And please note here, you may be wondering how it knows which side, but there's actually, in this complex, there are chemical signatures by which it can tell which strand is the new and which is the old. So it will take out the base that's incorrect in the brand new strand, assuming that the template strand is correct. So pretty clever. And that is the end of our DNA replication video for today. Can you please complete the sheet, including the table that's included summarizing what all of the players do in replication? And please bring any questions that you have to class on the assigned day.